Welcome to the Chapter 10 podcast over photosynthesis. Today specifically we're going to go over some of the kind of overview topics about photosynthesis, and we'll try to get through the light reactions. We'll then pick up with the light independent reactions, Calvin cycle, whatever you want to call it, with the second podcast as we wrap up. So getting started, we'll see here on the front we've got the equation for photosynthesis. Now, if you guys remember cellular respiration, you'll see this is pretty much the opposite. So with cell respiration, we started off with our reactants being a glucose and oxygen, molecular oxygen. And then we reacted to form CO2, H2O, and energy in the form of ATP was captured. In this case, we are going to capture energy in the form of light to allow us to build, to do this endergonic reaction of taking CO2 and H2O and assembling them into a sugar and molecular oxygen. So this will be kind of the exact opposite process. It's just the energy is a little bit tweaked, where in one of them we're storing energy for later use, ATP by the cell, and in one of them we're grabbing energy from light, which we will use to make ATP, but that energy will not be used elsewhere in the cell. It'll stay in the chloroplast or the thylakoid to specifically allow us to build up the molecules, because anytime you're building, anytime it's anabolic, it's going to take energy. So even though we will bring up stuff like ATP here, just keep in mind, this will not be ATP used elsewhere in the cell. This is only going to be produced and used locally. Don't misconstrue this with what a cell has to do to get ATP to power the cell. Now, when we look at this, the common term for things that are going to do photosynthesis will be autotrophs, or producers. You might see that term. And I want to make sure you realize that plants are what most of us think about, but there are things like brown kelp you can see here. There's different types of cyanobacteria that will go through and do this, as well as different types of algae. So most of these are eukaryotic with the exception of cyanobacteria. Those are going to be regular bacteria. But I want you to realize that it's not just plants that do this. There are ultimately prokaryotes that can do this. There are eukaryotes that are fairly simple to fairly complex that are not plants. The algae go anywhere from single-celled to, in some cases, you're talking hundreds of feet tall if you're looking at the brown kelp forests in the ocean waters. And then we obviously have our plants. So don't just kind of think that this immediately must be plants. There's a lot of stuff that can do photosynthesis. And the idea here with autotrophs and photosynthesis is they are going to use inorganic molecules, all right? So this can be things like CO2, and they're going to build organic molecules from those inorganic ones. And these organic molecules will then be used for energy, so essentially the whole food idea you see people say, as well as for structure. When they want to go through and build a new cell, when they want to have more of a cell membrane, all the proteins, all the lipids, all of the carbohydrates, all the nucleic acids that they need, they're going to have to assemble. So this idea of photosynthesis, even though we oftentimes use glucose as that poster child again, acting like that's what we produce, you'll see that we don't actually produce glucose necessarily. We'll produce a smaller molecule, G3P, which we can reassemble. I shouldn't say we, but plants can, and autotrophs can reassemble to form any molecule they need. So this is going to account for all the organic molecules that you're going to typically find in any autotroph. Is going to be done through one of these processes, one of which we're going to talk about, the other one I'll just briefly mention. So photosynthesis is the one that we're going to talk about more, and that's the one that's going to do this using light as energy. Whenever you want to build these structural or storage molecules, you have to have an input of energy. So if you're talking about photosynthesis, this will use light. There's also chemosynthesis, which is going to use chemicals. So typically it can be hydrogen gas, it can be hydrogen sulfide. There's a variety of different versions of this, but these guys tend to be much fewer in number. They tend to live in kind of extreme scenarios, like the deep sea thermal ocean vents that you'll find. They're not your everyday. So most of the time when we're talking about autotrophs, probably well over 99% of the time, we'll be talking about the photosynthetic guys. So that's what we're going to focus on here. Later on, when we get to prokaryotes, we will bring up chemosynthesis a bit more, but just keep in mind there isn't just one way to be an autotroph, it's just there's one really common way, so we're going to focus on that one. And then heterotrophs, this is going to be stuff that needs to ingest their organics. So they're going to get their organics from either eating them, or in some cases it could be absorption, the idea that they can kind of live in an area where they've digested stuff, so it's next to their cell, and it's in small enough pieces they can bring it directly across into the cell. So they don't have to go through and wholesale eat it like an animal would. But these guys have to get it ultimately from somebody else. We're heterotrophs. We can't make our own food. We can't make our own structural organic compounds. We go through and we eat other stuff, and we ultimately use their organic compounds to allow us to make what we need. Now, the chloroplast is going to be the site, especially in eukaryotes, or specifically in eukaryotes, where we're going to see photosynthesis occur. So this one's good to just get a basic reminder about the structure of it. So the chloroplast is going to be colored. You'll usually see it colored green because the most common pigment that you'll see is going to be chlorophyll A. There's also chlorophyll B that's pretty common. In some things like algae, you will see that it might not be green. It could be more of a yellow or a brown or a red. It all just depends upon which pigments are most common. Most plants tend to have more chlorophyll, and chlorophyll tends to be green, so that's why you're going to see this idea of the green chloroplast is the classic one. You're going to see inside of this, beyond the double membrane, you can see there's these two membranes. This will not be like the mitochondria, though, where the inner membrane was where it happens. It's going to actually be these little membranous sacs that there are inside. So these little membranous sacs are ultimately going to be the thylakoids. Right? So you can see thylakoid. That's one there. There's actually a stack, so there's about 5 of those. And if you ever see a stack, the whole stack is typically called a grana. So if you see somebody bring that up, that term grana, just remember it's just a series of thylakoid stacks. And that's the ultimate site of where most of this stuff is going to happen. So just like if you looked at a mitochondria, it was really the matrix and the cristae where most of the stuff happened. When you're talking about the stuff here, it's going to be the thylakoid, and then that space, that fluid-filled cytosol equivalent, if you will, that's outside of the thylakoid is called the stroma. So the stroma is going to be kind of all this stuff here, this darker green. As I said, it's kind of like the cytosol or the cytoplasm stuff that is adjacent to and touches up against that thylakoid membrane. Because you'll see there will be this idea of transferring some stuff from one side of that membrane to the other, just like we had in the mitochondria, where we transferred H+ from the matrix to the intermembrane space. You don't want to cross that cristae. Some other ideas, mesophyll, this is just a name for leaf cells, essentially. So when we talk about where this happens, it's usually going to be, at least in plants, in the leaf cells, because those are the ones that are exposed to light. You'll see much of the plant is not necessarily green, especially things like trees. And so it's going to be the parts that are exposed to light that are going to typically have these chloroplasts, because if you give chloroplast to your roots or something like that, it just wastes your energy. And so plants have evolved to kind of be efficient, and so they tend to have a lot of chloroplasts that are smashed into these mesophyll cells, but not so much other cells. And the stomata, when you look at a leaf, so if I kind of draw a horrible leaf here, okay, so you're going to have these cells here, going to have a bunch of chloroplasts, but they'll also have these kind of little pores, and we call those stomata. They're typically on the bottom of the leaf to try to prevent too much evaporation, because you'll see these pores are what the plant will open up, and it allows carbon dioxide gas to get in, which we need for photosynthesis, right? We need water, and we need ultimately CO2. Now the problem for a plant that we'll discuss more later is that when they open up these pores, it lets them get CO2, but it also allows water to evaporate and leave, what we call transpiration. So if we see me talk about like this gas exchange where oxygen can leave, carbon dioxide can come in, or if I talk about water issues where you have water leaving that can become a problem for the plant, that's going to go back to these stomata, these pores that the plant has to allow them to get what they need, but there will be consequences for that. Okay, an overview. This will involve redox reactions ultimately when we do this. You'll see specifically we have NADP+, which should strike you as familiar to somebody else, NAD+, that will become NADPH. So instead of NADH, it becomes NADPH. And how I like to remember this is the P is for photosynthesis. So if you're never sure, should I say NADH or NADPH? I always make sure that if it's photosynthesis, the P is there. The P is for photosynthesis. If there's any other way you can remember it, just that's fine, but try to do your best to have some way of getting through this. You'll see that we have two big parts. We've got the light reactions and the Kelvin cycle will be our two big chunks we're going to focus on the light reactions today. And what's going to happen in the light reactions, as a quick overview, is we're going to take water and we're going to split it, which is called photolysis, using light to split something, in this case water. You might see them call it hydrolysis as well, that's splitting water. And that's ultimately where the oxygen that we give off, that we breathe, comes from. So if you see them talking about like where does the molecular oxygen released by a plant come from, it comes from splitting water. And we'll split the water so that we can get electrons, because the electrons are what we're going to do with this whole light reaction process. The light reaction process is going to be fairly similar to the electron transport chain. And the products of the light reactions will be ATP and NADPH. Okay, they're going to be the energy molecules, because we need these energy molecules to let us do the Kelvin cycle, which is where I take 3 CO2s and convert them into one 3-carbon sugar called G3P. So this is our end goal. This is really what we're after in photosynthesis, is to produce this one G3P, this one 3-carbon sugar. But in the process for doing that, we need the CO2 to get carbon, we need the light reactions to give us the energy molecules, ATP and NADPH, and we have to split water ultimately to get the electrons so we can do this process. And then light, of course, provides the ultimate source of energy for the whole process, because the light energy is what's actually allowing us to make ATP and NADPH. We're not breaking down food to do this. We're capturing light and using its energy, specifically energy caught in an electron, to let us do this process. So you can see the products here, and the products that we produce in light reactions will be the reactants that we need to do the Kelvin cycle, where we can take the CO2, take some H plus that we've gotten by ripping water apart, and some electrons that we've also essentially gotten by ripping water apart, and stick them together to make a sugar. In this case, like I said, it's going to be that one G3P guy, a 3-carbon sugar, that we can then make into whatever we want. If we stick 2 of them together, we get glucose, but we can also switch it all around and make amino acids, to make fatty acids, whatever the plant needs. Okay, the light reactions are going to take place in the thylakoid membranes. So you'll see this is one of the reasons that a chloroplast tends to have lots and lots of thylakoids, where they're in stacks inside of it, because it gives them more surface area. The more surface area they have, the more enzymes, the more of these what we'll call photosystems can be embedded. And these photosystems, just like the cristae was folded to allow lots of electron transport chains, these photosystems are going to act a lot like an electron transport chain, where we're going to use them to take energy that's stored in an electron, specifically an electron that's been hit by a photon of light. So ultimately it's got this stored electron. We call that an excited electron. So once light hits the pigment that contains the electron, excites the electron, we're going to put it into this photosystem, and you'll see the reaction center is just a chlorophyll A molecule. It's a specific pigment that's going to pass on that electron to something called the primary electron acceptor, which is just the first guy in the photosystem. So the photosystem is going to have these cytochromes, just a fancy term for type of protein, that much like the ETC is going to go through, and it's going to extract the energy from that electron bit by bit, and use the energy it extracts from that electron. So if I draw kind of a crappy version here, so as the electron kind of falls down through this process that is the photosystem, it's going to allow it to pump H+. Now in cell respiration they pumped out. In this case they're going to pump H+ in, and so we'll see that the concentration of H+ is going to be highest inside that thylakoid. So that means it's going to be most acidic inside the thylakoid, and the stroma itself will be more basic. And then we can allow these H+ to diffuse back out through an enzyme that we will call ATP synthase. Same guy as we had before, same basic idea as we had before. So if you guys remember the electron transport chain, if you remember chemiosmosis or oxidative phosphorylation, or whatever you want to call it, this is going to be the same basic idea, with the only difference being it's occurring in the thylakoid membrane, not the cristae, and it's pumping the H+ in this case, instead of out. So we've just kind of turned around these cytochromes to make them go in the other direction. So that's pretty much it as far as an overview of the light reactions. Now the light reactions themselves are going to be largely dependent upon what pigments you have, because the pigments are what catches the light. They're the ones that can capture it. When they capture it, their electrons jump up energy levels. That's how they store energy. They can then take those excited electrons and pass them to that primary electron acceptor. They can pass them to the photosystem itself. And so each pigment is going to have a different range of wavelengths or frequencies of light that they're able to capture. They can't just get any light. And so you'll see the common ones are going to be chlorophyll A and chlorophyll B. And both chlorophyll A and chlorophyll B tend to be strongest in terms of absorbing this reddish light and ultimately this bluish to violet light. It's kind of like the bluish end of violet, as well as like the I don't know, I guess pretty much most of the spectrum of blue, except for the greenish blue. And so this is one of the reasons that you'll see plants look green, because they're not absorbing this stuff in the middle, so this greenish yellowish stuff is the ones that get through. And so a plant that has lots of chlorophyll will typically look green. Now there are other guys called like carotenoids, because they tend to be a class that is largely like yellows, more like reds. In some cases you'll get obviously orange, the whole carotenoids, carrot. And so you'll see that these guys will absorb different wavelengths, so they'll be different colors. And a cool way of seeing that most plants have a lot of these guys is if you look around in the fall, the leaves haven't started changing yet. But when you start paying attention, you'll see as plants go dormant, as trees go dormant, they break down their chlorophyll, but they tend not to break down their accessory pigments, their extra pigments, those carotenoids. And so you'll see whatever ones were there the whole time, you'll see them by themselves during the fall. And so you'll see lots of leaves will turn yellow or orange or red. And what you're seeing is these carotenoids that they use to be more efficient, because that allows them, by having different types of pigments, to absorb more wavelengths of light, what we call the action spectrum, the specific wavelengths of light that let it function and complete photosynthesis properly to do the light reactions. So just keep in mind this varies from plant to plant, but because that reaction center, that one main pigment that passes off the electron, is going to be chlorophyll A, typically everybody has at least a little bit of chlorophyll A, but they can have that chlorophyll A that passes off the electron, if you will, to the primary electron acceptor. They can have that one that's kind of in a sea of other pigments that don't have to be chlorophyll A. And so if one of these guys gets their electron excited, say this guy here, they can have that electron kind of just jump until it gets to the reaction center and sends the electron off. So this will be how the process works. And then the Engelmann's experiment, just a real quick thing, this is what demonstrates that you have this idea that red and blue light especially tend to be the most effective. This is where a guy grew algae, and he grew aerobic bacteria in the container with the algae. And so around the algae that were exposed to red and blue light, you saw the most aerobic bacteria that were living. And this was because it was at those areas that there was the most amount of oxygen being produced, which is what the aerobic bacteria needed to properly get their energy. And so this is another way of kind of observing in a very simplistic way, without needing a lot of high-tech equipment. You can just shine a light through a prism that gives you a spectrum of light that's kind of shining down on this algae, so that different parts of the algae get more or less, and you can just see where those aerobic bacteria go. And in this case, they'll always cluster around the areas in the action spectrum that are ultimately most able to cause photosynthesis to occur. And that is the blues, specifically towards the violet end, and the reds. Now there's two basic ways of kind of going through the light reactions. The first is going to be called non-cyclic electron flow. And that means you're going to go through both photosystems, because there's two photosystems. Now you're going to be annoyed because they're not in order. They discovered photosystem 1 first, but then they found out later it was the second photosystem. So rather than renaming them, they just went with it. So technically photosystem 2, which I'll commonly write PS2, comes first. And then photosystem 1 actually happens second. But it's the same idea for both of them, where there's going to be a bunch of pigments here, an electron's excited and dumped onto the photosystem, where it goes through a series of these cytochrome proteins, and energy is extracted that allows us to pump H plus in, as you can see. So H plus is coming in. Now that electron, we took it from the pigment. So this pigment is now down an electron, and we have to give those electrons back. So what's going to happen is we're going to split water, so H2O is going to ultimately break apart, and it's going to become an O2. This is the gas that's ultimately given off, so this is the waste oxygen that we'll see released. It's also going to produce 4 H pluses, which are useful because we can pump them. We can also use them to assemble sugars later. and it's going to produce 4 electrons for every 2 waters we split. And these electrons are ultimately what's able to be given back to the pigments that are at the start of photosystem 2. So this is how we donate the electrons back. So photolysis is ultimately what provides the electrons for the pigment, so the pigment can donate electrons to the PS2, and then once we're done with photosystem 2, you'll see at the end of photosystem 2, we dump the electrons onto the pigments that are at the base of photosystem 1. And so that allows us not to have to worry about giving more electrons to photosystem 1, because they get the electrons from photosystem 2. We don't have to split water again. In photosystem 2, what's normally going to happen is we're going to get an excited electron, take the energy from it, but in this case we're going to use it to take NADP plus and convert it to be NADPH. So we're going to use it to make NADPH to reduce that, which is going to allow us to now have energy stored in this electron carrier, much like we did in cell respiration. So it's just the electron transport chain obviously just made ATP. Here we're going to make ATP with photosystem 2, with the idea being that photosystem 2 is pump this H plus out. We can then allow the H plus to go through ATP synthase just like before and make ATP. So when we're talking about this, photosystem 2, ATP. Photosystem 1, NADPH. That's their job. At the end of this, the electrons that we have are dumped on NADPH after photosystem 1, so those electrons are now good to go. We're fine. You know, photolysis gave us electrons, it went to the pigments, they got excited, to PS2, to PS1, eventually on NADPH, so we're all good. And now NADPH and ATP are ready for us to use when we get to the next phase, which will be the Calvin cycle. But there is one issue. In general, we need more ATP than we need NADPH. And non-cyclic electron flow is going to make equal amounts of ATP and NADPH. And so that causes us an issue. We don't want to have where we're just rolling in NADPH, but we have a shortage of ATP. So what we can actually do is when you go to photosystem 1, you'll see Redox, and you don't have to memorize that, but this FD guy, he's actually capable of going either direction. So he can kind of go to the right, as you see here, and make NADPH. That's one way. That would be non-cyclic. But he can also go this way and dump it onto the cytochrome and kind of use the same machinery that we have in photosystem 2, except in this case, we're doing it with an electron that was excited by photosystem 1. So we'll say that we're going to do photosystem 1 only, but photosystem 1 is going to work in the same way photosystem 2 does, in that we're going to dump it on the cytochromes, it's going to pump H pluses in, the H pluses can then go through ATP synthase, which is going to allow us to make ATP. And we can keep doing this and doing this, so as we get low in ATP, we can use that kind of feedback inhibition process, whereas we get low in ATP, we can do cyclic electron flow and make a bunch of ATP by just having things go left, if you will, by having ferredoxin just drop off the electron at the cytochromes instead of dropping it off where it's going to make NADPH. And so this allows us to balance things, and when we get enough ATP, we can just have ferredoxin go use the other path, the non-cyclic path, which generates NADPH for us. So that would be called cyclic photophosphorylation instead of non-cyclic photophosphorylation. And the whole photophosphorylation is just using light to make ATP. So all that matters here is which one's doing it. Is it photosystem 2 that's non-cyclic? If it's photosystem 1, it's cyclic. In general, though, it's the same process. It's the same guys, really, that you're using to do it. It's just a matter of did the electron come from photosystem 1 or did the electron come from photosystem 2? That's the only thing that's changing. But this flexibility allows for them to deal with the fact that they want to produce more ATP than NADPH.